Okay, it sounds like everyone's logged in, so we're going to go ahead and get started. Thank you so much for joining our webcast today on the ProStorm Cytokine Release Assay. This presentation will be interactive, and at the conclusion you'll be able to type any questions you may have into the question and answer box that should be on the right side of your screen. If not, there is an icon at the top of the screen with 4 arrows that you can shrink down or expand your slides. So if you don't see the Q&A box, just shrink that down and it should appear. So this presentation is also being recorded and will be available on our webpage. So if any of your colleagues were unable to attend this live presentation, then we certainly invite them to listen to the recorded version online. My name is Dr. Emilee Knowlton, and I'm an immunology sales specialist with Proimmune. And as you can see, the focus of today's webcast will be on our ProStorm Cytokine Release Assay, a risk mitigation tool for first infusion reactions. So I'd like to begin by first introducing the topic of first infusion reactions, which are really innate responses that can occur very rapidly in response to biotherapeutics. There are many types of infusion reactions. For example, tumor lysis syndrome, which is the most relevant to something like cancer chemotherapy, in which the therapy is delivered to the patient, and following the targeting and killing of the tumor cells, there's a massive release of cytokine and other material into the plasma and surrounding tissue. Other examples include complement activation-related pseudo-allergy reactions, and type 2 hypersensitivity-like reactions, and of course cytokine release syndrome, which will be one of the focuses today. First infusion reactions are typically dependent on the interaction between the biologic and the target, or the innate immune system. And actually drugs that target membrane-bound antigens or receptors tend to carry a greater risk of inducing cytokine release than those that target soluble molecules. A systemic inflammatory response can occur due to first infusion reactions, and those symptoms can range quite significantly from mild to even fatal. And these can include things like fever, skin rash, hypotension, and cardiac arrest. First infusion reactions, however, are quite common with delivery of biotherapeutics, and over time clinicians have been able to mitigate some of these side effects using various techniques that I've listed here at the bottom of the slide. And these can include things like administration of a lower dose of the biotherapeutic, a slower infusion rather than a rapid injection, and then the use of an antihistamine or corticosteroid in conjunction with the biotherapeutic. So these methods have been known to alleviate some of those symptoms. So as I mentioned, first infusion reactions are quite common, and many successful drugs that are on the market today do have a high rate of first infusion reactions. And some of those are listed here. The first in this table is Campath, which is an anti-CD52 antibody that was indicated for MS treatment. And it's known that a very strong cytokine storm occurs in about 100% of the individuals receiving this therapy. But again, these symptoms can be somewhat mitigated through the means I just mentioned. Remicade and Humira are both anti-TNF-alpha therapeutic antibodies that are used to treat rheumatoid arthritis. Remicade is a chimeric antibody, while Humira is a fully human version. And these antibodies have been observed to induce incidence rates between about 6 and 10%, so still somewhat frequent among patients receiving these therapies. Herceptin is an anti-HER2 antibody, and around 40% of individuals receiving this treatment will have a reaction, while Rituxan is an anti-CD20 antibody, and almost just around 3/4 of people who are receiving this therapy will develop a reaction. So again, many of these drugs do have a high rate of first infusion reaction, so that factor doesn't necessarily mean the biologic shouldn't move forward, but ultimately you would want to identify the leads in the preclinical phase that have a lower potential risk of cytokine storms. So the last featured antibody on this list is TGN-1412, a humanized anti-CD28 agonist monoclonal that was developed by a company called TGenero. And this antibody also induced reactions in 100% of the patients, but unfortunately the symptoms were much more severe and is a key example for biotherapeutic inducing severe cytokine storm. TGN-1412 was in a clinical trial in London, so in the United Kingdom, in 2006, and it was originally intended for B-cell chronic lymphocytic leukemia. And TGN-1412 was administered contemporaneously in all 6 patients, and then all of them were actually hospitalized, and 4 of them experienced multiple organ failure. And the dose that was administered to the patients in the clinical trial was actually much less than what was tested in the toxicology animal studies. So the adverse reactions were not anticipated at all. And this is a key example of how the animal model was not a suitable comparison for what would occur in humans, as the mode of action wasn't replicated. So this certainly highlights the need for an in vitro human assay to assess such risk. So using human in vitro assays to understand the potential risk of a biotherapeutic could obviously be very beneficial to a company. And there are now key regulatory considerations to bear in mind. In 2014, the FDA released a draft guidance on immunogenicity assessment for therapeutic protein products, stating that data from animal studies should be supplemented by in vitro assessment of cellular activation, including proliferation and cytokine release in human whole blood or PBMCs, and that the measurement of a cytokine panel should be as broad as possible and include IL-6, interferon gamma, and TNF-alpha. So this brings me to the ProStorm Cytokine Release Assay, which was developed by Proimmune as a hazard identification tool to identify first infusion reaction risk for a biologic. The assay is a fresh whole blood assay with minimal test interference, and in fact the donors that are recruited for each project are done so specifically, and once the blood is drawn, it immediately goes into the assay. So there's no additional processing or manipulations which could further stimulate the cells. And I'll describe the assay workflow more in just a moment. But typically the studies done for ProStorm range from 20 to 50 donors, which is really important because variability among donors is quite large. And as we saw in the previous table, many patients receiving biological therapies will mount infusion reactions, but the degree and severity will also vary. And also, of course, the same biological molecule will not necessarily act the same in every individual. So having access to a large panel of donors is really the best approach to assessing the risk. So the donors used in the study are recruited by Proimmune through an online registration. These are all local donors. The lab operations are held in Oxford in the United Kingdom. And these healthy individuals have been identified as free from symptomatic viral and bacterial infections. They haven't taken any steroidal anti-inflammatory medications for 7 days prior to the blood draw, or any non-steroidal anti-inflammatory meds for 3 days. They also must not be taking any blood thinning medications or antibiotics. So the recruitment stage is quite a streamlined process and typically takes a maximum of 3 weeks for a 40 to 50 donor study. So the workflow following donor recruitment is shown here. So the blood is drawn into heparin tubes and then plated for the assay. And each test article is added in 12 pliquets, so 12 wells, in a range of doses that you would expect to see in patients. So this typically ranges between 0.1 to 100 micrograms per ml, and we typically do 4 different doses. And because the blood is not diluted, again, minimal interference, this does require that each drug be provided at 50 times the highest desired final drug concentration. So this is certainly something to keep in mind when designing the study. So the blood and test articles are then incubated overnight, and the blood plasma is separated and then analyzed on our Prorate Ultra platform, which I'll describe more of in just a moment. The cytokines that are quantified are done so on that platform, and they include interferon gamma, TNF-alpha, and then interleukins 2, 4, 6, 8, and 10. And so this panel obviously includes markers which are typically seen in a cytokine storm. And then following the analysis of the data, the results are reported and a project debrief is provided with our assay scientists. As I've mentioned, the Prorate Ultra platform is used for cytokine quantification, and this is a technology that was developed by Proimmune for detection of protein-protein interactions as well as protein-peptide interactions. And it's a very flexible, customizable microarray platform which can be used to create up to 24 subarrays, which you can see in the slide on the right, and that can assess up to 30,000 different features per slide. This technology has quite a few applications, one of which is linear B-cell epitope mapping, but also for cytokine quantification for the ProStorm assay, which we're discussing currently. So it's essentially set up as a miniaturized ELISA, where we print a cytokine capture antibody onto each of the subarrays, and then that's followed by the plasma sample or a standard curve sample containing the recombinant cytokine. Then followed by the second layer, which is the cytokine detection antibody containing a fluorescent detection label. So this method is highly sensitive, and when compared to ECL ELISAs, you actually get a higher sensitivity with a wider dynamic range, and it's very reproducible. One of the really nice features, actually, is that it requires a very low sample volume, so only 1 microliter, which makes it quite efficient to analyze large number of samples, making this a high-throughput system. Okay, so now I'd like to share some example study data that was performed here at Proimmune. And in this study, we recruited 30 donors, and the test articles we included in the project were Remicade, which we've already discussed, which is an anti-TNF-alpha antibody, aggregated Remicade, which was the Remicade stir-stressed over a 24-hour period, which caused a heavy aggregation determined by visual inspection. We also looked at Herceptin and Vectibix, which are both anti-EGF receptor antibodies. Herceptin is a chimeric antibody, and Vectibix is a fully human version. And with any ProStorm project, we also suggest including drug assay controls or comparators to run alongside your test articles. And this can be any number of things. It may be drugs with a similar mode of action, a drug with the same target, a biosimilar or variants within your own development program that can be used as benchmarkers, or really any other molecules that you're familiar with in your development process. And in the study I'll describe now, we used Campath and Erbitux. As you saw in the previous slide, Campath, the anti-CD52 antibody, is associated with cytokine release syndrome and was used as our high response comparator, as it has a characteristic response for interferon gamma, IL-6, and TNF-alpha. While Erbitux, or cetuximab, was used as the low response comparator. And this does have a relatively low incidence of first infusion reactions unless there is a pre-existing sensitivity to a particular sugar residue, which can be the result of a tick bite, but this is quite rare. So in addition to the drug assay controls, we also run internal assay controls, and PBS serves as our negative control. And PBS responding donors are considered outliers and excluded from the final analysis. And for all of the other donors, we subtract the PBS responsiveness. Staph endotoxin B, which is known to induce a strong interferon, gamma, and other cytokine response, is used as our overriding positive control. So the analysis for ProStorm is described here in more detail. As I mentioned, we do exclude donors who respond highly to PBS. And in this example study, one of the donors was excluded, leaving a total of 29 donors for analysis. Again, the Prorate Ultra platform was used to quantify the cytokine concentrations for both the samples as well as the standard curves using the recombinant cytokines. And then the data that was obtained for Erbitux was used to set a 95th percentile threshold across the increasing drug concentrations, which you'll see an example of in just a moment. but this means that 95% of the donors who did not respond to Erbitux were used to create this threshold. This is really just an example of how the comparators could be used and how we assess the data. Certainly this is flexible and can be done using other biotherapeutics, but ultimately the cut points are likely drug and dose dependent, and the data should really be viewed as a whole. Okay, so now on to the data generated from the 29 donor study. In the report, you will receive a graph for each cytokine against each of the test articles. And in this example, we're looking at the IL-6 response to Remicade. So shown across the top is are the increasing drug concentrations from 0.1 to 100 micrograms per ml. Beneath each one of those concentrations are the responses for that drug dose. And then across the x-axis are the cytokine concentrations in picograms per ml on a logarithmic scale. So within each graph, you'll see the red line I was describing, and again, this is set using a 95% threshold from Erbitux. So if we start on the far left at the lowest concentration at 0.1 micrograms per ml, you see there's only one donor responding, which is indicated by the filled-in blue diamond, while the other 28 were considered non-responders. As we increase the drug dose up to 100 micrograms per ml, you see the number of donors responding or producing IL-6 increases to 6 out of the 29, or 21% of the study participants. So when we look at the IL-6 response of the different test articles, as a whole, we can see that there is quite a range in both the quantity of cytokine production and the number of donors responding to that particular drug. So on the lower set of graphs is Erbitux, the low response comparator, and in the upper right corner is Campath, again our high response comparator. And as you can see, at the 3 highest drug doses for Campath, the IL-6 responses were quite high and considered positive in 78, 97, and 96% of the donors. So you can see that this comparator works really quite well in the system and is consistent with what has been detected in the clinic. As for the test articles, the upper left set of graphs is Remicade, which we've just gone over in the previous slide, and beneath that is aggregated Remicade. So there's very little activation occurring at the 2 lower doses, but as soon as you get to 10 micrograms per ml, it's really in the same level or ballpark really as Campath. So you can see that the innate immune system can really respond to aggregation and recognize aggregated proteins as a risk factor, which suggests that the quality of the drug is quite important, but also the mode of delivery. On to the other 2 test articles, Herceptin and Vectibix, which were on the bottom right. There's a very observable dose response seen for Herceptin with 0, 7, 14, and then 28% of donors responding or producing IL-6 in response to Herceptin, while Vectibix just had responding donors of around 17 and 14% at the 2 highest doses. The data is also presented in the report with the mean value for each concentration for each drug. So here again you can see a very nice dose response in Campath with the mean levels indicated by the red dash, and also elevated levels of IL-6 for aggregated Remicade at the 2 highest concentrations, which is very comparable to our positive control, the staph toxin. So I've only shown the data for IL-6, but we did detect a good correlation between the in vitro study data and clinical observations, not only for IL-6, but also IL-8, interferon gamma, and TNF-alpha. We also saw that our high and low dose comparators, Campath and Erbitux, were consistent with what has been seen in the clinic. And so using these comparators or other benchmarks in the ProStorm assay, you'll really be able to define or scale the relative risk of your biotherapeutics. So to summarize, the ProStorm assay is an assay for hazard identification and risk mitigation. So I'd really like to stress that it's not a predictive test that will describe what will ultimately happen in the clinic, but rather a research tool. It is a simple whole blood assay that minimizes test interference and provides a flexible study design. So the assay can be tailored to address particular questions surrounding your biotherapeutic, like the mode of action and ranging drug doses, and can be assessed for a large number of cytokines in a very large donor cohort. And because these donors are recruited specifically for each project, this may allow for follow-on testing if necessary. And in some instances where disease-specific donor cohorts are required, then we can certainly try to accommodate that as well. And we know that cost and turnaround time is also very important when doing preclinical assessments. So we strive to make this a cost-effective and time-efficient project, providing results for 40 to 50 donor studies in just 6 to 8 weeks. So today I focused on first infusion reactions, and the ProStorm assay is a tool to assess that potential risk, which again is really more about innate responses. And I'm not going to go through this entire cartoon, but as you can see, the process leading to unwanted immune responses can be quite complex, and there are just many points along the adaptive immune response which can also be assessed for risk, and those are highlighted here. And we have multiple assays to look at the interaction between antigen-presenting cells and T cells, as well as assays and reagents to assess B cells and antigen-specific T cell responses. So as we're constantly striving to come up with new targets and technologies and biotherapeutics, the risk for unwanted immune responses certainly increases, and evaluating that risk for immunogenicity in the early development process becomes essential to the drug's success. So of course, no one assay will answer all of your questions, and so the platforms that we can provide can be integrated to address the specific needs of each project. Summarized here is what we consider our immunogenicity toolkit, which highlights several of our immunogenicity assessment assays and their applications. And we're happy to work with you and discuss program needs and questions to best select the appropriate tools to answer those questions. And the assays in this toolkit include our reveal binding assays for determining on and off rates of peptide binding to MHC class 1 or class 2 molecules, the naive T cell proliferation assay and DC T cell proliferation assays to determine CD4 responses to peptides and whole proteins, the ProPresent antigen presentation assay for determining naturally processed and presented class 1 and class 2 epitopes, and then the ProStorm assay, which I've described today. In addition to our immunogenicity risk assessment platforms, we also have several supporting technologies and assays, including our Pro5 pentamers for detection of antigen-specific CD8 T cells, our Pro-M2-MHC monomers, which we have recently launched and can be used for detection of antigen-specific CD4 T cells. We also have an alpha-gal ceramide-loaded CD1D tetramer for identification of NK T cells. We can provide custom peptide synthesis as well as pre-mixed peptide pools as a part of our Think Peptide brand. And we have several assays that can be used in immune monitoring programs such as ELISpots, ICS, and phenotyping. So I mentioned earlier that we do source donors specifically for each ProStorm study so we can acquire fresh blood. But for the cellular in vitro immunogenicity assessment assays I just mentioned in the previous slide, we also provide a donor cohort program. And these are pre-stocked, cryopreserved PBMCs from hundreds of donors that we've typed for class 2. And we'll select a cohort of donors for each study to represent the global population of HLA expressions. HLA typing is quite an invaluable tool for study design and patient cohorts, and we also offer this as a service for submitted blood, PBMC samples, genomic DNA, or saliva samples. So Proimmune isn't your typical CRO. We would really prefer to think of ourselves as a strategic partner that can offer support through our experience and technical competence. We are constantly striving to understand immune responses through improvement of our products and services based on the best and newest technologies. We were founded in 2000, and as I mentioned, our headquarters and lab operations are located in Oxford in the UK. And we have customers really all over the world, essentially wherever immunology research is being conducted, and some of our publicly disclosed clients are shown here. So I'd just like to end the presentation by briefly mentioning our upcoming Mastering Immunity Conference that will be held in Boston at the British Consulate this September on the 14th and 15th. It's a workshop-style format with a strong focus on correlating data from preclinical predictive tools to clinical outcomes, as well as discussions surrounding the benefit of quality by design and the effect that has on modulating immunogenicity. So we will be hosting several wonderful speakers, and there's an updated list on our website. Some of our past speakers are shown here, and their talks can also be found on our website. So I certainly invite anyone who's interested in this to please check out the website and register to attend. Okay, so that concludes the presentation on ProStorm, and I'm happy to take any questions. Please just type them into the Q&A box that's on the right of your screen. And for anyone who dialed in late, if you don't see the Q&A box, there's an icon at the top of the screen with 4 arrows, and you can shrink down your slides and pull that box up. So while we're waiting for questions, here we have a first one already. One second. Okay, so the first question is, what is SEB? So we use this as a positive control in the assay. This is staph endotoxin B. It looks like we have a few more questions coming. So while those are being typed in, if anything pops up in your head later in the day and you don't think any questions right now, you can reach me at eknowlton@proimmune.com, or you can contact our office directly at inquiries@proimmune.com. And of course, our website is listed here as well. So this question is, at what stage of the assay do you do linear B-cell epitope mapping? My apologies if I made that confusing. The Prorate Ultra platform that we perform the cytokine analysis on and use to quantify the cytokines is quite a flexible tool. So it has other applications like linear B-cell epitope mapping, but that's not actually part of the ProStorm assay itself. So I mentioned that kind of as an additional application, but my apologies if I made that confusing. Okay, this next question is, if I would like to assess whether a peptide binds to both human MHC class 2 and murine MHC class 2, can you help me? Yes, we can do that. We do have binding assays available for both human and mouse, and we can connect after this, and I can let you know which alleles we have available. We have 56 different class 2 alleles available for human and some for mouse as well. So I can provide you with some sample reports, and we can discuss project specifics. So just feel free to contact me using that email. Okay, let me see if there's any other questions. We're still waiting on a few more to be typed in. And again, this presentation was recorded, so it should be on our website within the next few days. So if you missed the beginning, if you didn't get dialed in and didn't have any audio in the beginning, then it should be available online. And we can also distribute a PDF of these slides as well. We also have sample reports available, which has a lot of this data that I've described in them. Okay, so the next question. Are you able to determine the level of response from your active cytokine release molecules, or can you only compare to the assay positive controls? Well, what you get are the actual concentrations of cytokines that are being released into the plasma, but having those quantities becomes more applicable when you have something to compare them to, really. So that's why we include those assay comparators. Of course, each comparator is probably going to be best suited if it's similar to your biotherapeutics. So that's why it's important to include possibly a drug with the same mode of action, or biosimilar or same target, so that you have a relative comparison of your therapeutic to another drug. Okay, we're just waiting on another few questions. Okay, do you conduct correlation between cytokine release and proliferation as well? We do not do that in this particular assay. We do have separate assays to assess this. The cytokines that are released are quantified as a part of ProStorm, and then we do have proliferative assays, so things where we're stimulating monocyte-derived dendritic cells with whole protein and then looking at the CD4 responsiveness to that, or stimulating PBMCs with peptides and looking at CD4 responsiveness to that. Okay, so this next question, the assay uses a soluble format. Yes, that's correct. Several reports indicate that the molecule must be immobilized in order to give a cytokine response. Can I comment? Yes, we certainly appreciate that there are several different formats of assays to test cytokine release, soluble versus immobilized, whole blood versus PBMCs. And really, I think it probably comes down to the particular drug itself. I don't think that one platform is really going to be able to address all of the different biotherapeutics that are out there, and it's best suited that you use the assay that's most relative to your drug. We haven't done any direct comparisons between our solubilized version or soluble version versus immobilized, so that would certainly be an interesting study. And these are really great questions. I thank you, everyone, for participating. So how does this assay compare to a blast? That's a great question, and I must say I'm unfamiliar with this. Can you elaborate on what this technology is? And while she's responding, basophilic activation. That's a great question. This is a basophilic activation. This has not been compared to that type of assay, so I can't really comment on how the results would compare. Just doesn't look like anyone else is typing in any questions right now. Again, if anything comes to mind, or if anyone would like to discuss, project specifics, pricing, anything like that, or have any specific questions, just feel free to email me. Again, the email is eknowlton@proimmune.com, and our office email is inquiries@proimmune.com. So you can contact us directly. We can provide these slides or sample reports or any type of customized proposal. So if there aren't any more questions, then I thank everyone for attending. I hope you found this very informative. And we do have other webcasts on our website. I invite you to look at those as well. Thank you so much, and have a great day.